Welcome to the deep dive. Today we're looking into something really fundamental, especially if you're heading into medicine, how your body actually powers itself.
Right. We're talking energy management.
Exactly. The intricate world of carbohydrate and lipid storage. How your system figures out what to burn now, what to save for, later. Think of your body maybe like a high-performance car, always needing fuel. But where's the tank? And how does it manage that fuel supply?
That's our mission today, really. To
Yeah.
unpack those major energy stores, how we build them up, how we break them down, and crucially, the regulatory systems that keep it all humming along.
Yeah.
And for anyone, starting medical school, getting these basics down is key for understanding so many diseases later on. We use some analogies, maybe some questions to make it stick.
Okay, let's start big picture then.
Yeah.
Where is all this energy stored? If you think about it like maybe a financial portfolio for energy.
Good analogy.
So in let's say, a healthy 70-kilo person, the vast majority, your kind of long-term savings account, that's triacylglycerol, fat, basically.
Yeah, loads of it.
Yeah, like 15 kilograms, holding a massive 600,000 kilojoules. Then you've got muscle protein, maybe 6 kilos. That's about 100,000 kilojoules, more like an emergency fund. You really don't want to dip into that if you can help it.
Definitely not ideal.
And then there's the quick cache. That's glycogen. Only about 0.4 kilos combined, but it gives you around 4,000 kilojoules ready to go.
It's a tiny amount compared to fat, isn't it?
It really is. And just for contrast, someone larger, maybe 135 kilos with obesity, their fat stores could be, 80 kilos? Holding like 3 million kilojoules? It just shows how energy-dense fat is.
It's incredible. But here's the interesting thing. Even though fat is the biggest vault, glucose is still the body's preferred fuel. And for some tissues, it's the only fuel they can use.
Like which ones?
Well, think red blood cells, white blood cells, the testes, kidney medulla, even parts of your eye, the lens, the cornea. They all run exclusively on glucose.
Oh.
And the brain. Well, stable blood glucose is just non-negotiable for the brain.
Yeah.
It needs that constant, reliable delivery. Like it has a VIP pass for glucose.
Okay, that brings up a really important question then, something every medical student needs to think about. If we only get glucose from our food for, a couple of hours after eating?
Right.
And tissues like the brain need it constantly, 24/7, how does the body keep that supply steady when we're not eating? Like between meals or overnight?
That's the million-dollar question, isn't it? How do we bridge that gap?
Yeah, let's keep that in mind as we go.
Yeah.
So the immediate answer, the first line of defense, seems to be that quick cache we mentioned, glycogen.
Exactly, your carbohydrate reserve.
So glycogen, it's basically a big molecule made of lots of glucose units strung together, and it's stored in little granules inside cells.
Highly branched.
Right, branched. I like the metaphor of a molecular tree. Glucose units are the leaves, linked together in chains, and then these branches sprout off. It all grows from a little protein core called glycogenin, like the root.
That's a great way to picture it. Those main chains are alpha-1,4 links, and the branches are alpha-1,6. Makes it very compact.
And where we find this glycogen matters, right? It's not the same everywhere.
Not at all. The roles are quite distinct. Think of the liver as the sort of central bank manager for blood glucose. It manages the supply for the whole body.
Okay.
Its glycogen stores, maybe up to 100 grams or so, get broken down into glucose, which is then released into the blood to keep those levels stable for the brain and other tissues.
Got it. Bank manager. And muscle.
Muscle glycogen is more like the personal trainer's private stash. It can store more, maybe 300 grams, but it uses that glycogen almost exclusively for its own energy needs during exercise.
So it keeps it for itself?
Pretty much. Muscle cells are missing a key enzyme, glucose-6-phosphatase. Without it, they can't release free glucose into the bloodstream. It stays in the muscle. That's a really critical difference.
Okay, that makes sense. So how do we build this glycogen tree? The process is called glycogenesis, making glycogen.
Yep, synthesis.
It starts by activating the glucose molecules, kind of prepping them. Then the main enzyme, glycogen synthase, links them into those chains. And another one, the branching enzyme, adds the branches.
Making it nice and dense.
Exactly. And this whole building project needs energy, right? ATP and another molecule, UTP.
Correct. It's an anabolic process, so it requires energy input.
And then when we need that quick energy boost, we need to break it down.
That's glycogenolysis, degradation, chopping down the tree, to use your metaphor.
Right. So the main enzyme here is glycogen phosphorylase?
That's the one. It snaps off glucose units from the ends of the chains, mostly as glucose 1-phosphate. Then a debranching enzyme comes along to handle the branch points, releasing a little bit of free glucose directly.
Okay.
But what's really vital to remember is that building it up, glycogenesis, and breaking it down, glycogenolysis, are not just the reverse of each other. They use different key enzymes.
Ah, okay. Why is that important?
Because using different enzymes allows for separate, independent regulation. The body can precisely control whether it's storing glucose or releasing it.
I see.
And this brings us back to your earlier question. When you haven't eaten for a while, the liver kicks glycogenolysis into high gear. It breaks down its glycogen and releases glucose into the blood. That's the first response to keep your brain happy. This usually lasts, about 8 to 12 hours.
Okay, 8 to 12 hours. That's our first answer. But, the body is so efficient, it seems wasteful to be building glycogen and breaking it down at the exact same time, like driving with the gas and the brake pedals pushed down together.
Absolutely. That would be a futile cycle, just
Yeah.
wasting energy.
So how does it avoid that? How does it make sure only one process is really active at a time? Is there like a master switch?
There is. It's called reciprocal regulation. Basically, when one pathway is switched on, the other is switched off. Think of it as like metabolic traffic lights.
Okay.
The key players are those two enzymes we mentioned, glycogen synthase for building and glycogen phosphorylase for breaking down. And the main controllers of these traffic lights are hormones. Glucagon and adrenaline, think stress or fasting hormones. They trigger a chemical change called phosphorylation. This phosphorylation slows down glycogen synthase, but speeds up glycogen phosphorylase.
So phosphorylation means break down glycogen, stop building it.
Exactly. More breakdown, less synthesis. Glucagon mainly acts on the liver, by the way. Adrenaline hits both liver and muscle.
And the opposite hormone, insulin.
Insulin signals the fed state. Plenty of glucose around. It promotes the reverse chemical change, dephosphorylation. This speeds up glycogen synthase and slows down glycogen phosphorylase.
So dephosphorylation means build glycogen, stop breaking it down.
Precisely. More synthesis, less breakdown. So you see, these hormonal signals act like very precise traffic lights, ensuring you're either making glycogen or breaking it down, but very rarely doing both significantly at the same time. It avoids those futile cycles.
That's clever. But sometimes things go wrong with these pathways, don't they? These glycogen storage diseases, GSDs.
Yes, unfortunately. They're rare genetic disorders where one of the enzymes involved in glycogen metabolism doesn't work properly. There are over 15 types, actually.
Wow.
For a clinician, understanding the basics is crucial. Take von Guericke disease, GSD type I. It's caused by a deficiency in that liver enzyme, glucose-6-phosphase.
The one muscle doesn't have.
The very same. So the liver makes glycogen, but it can't release the glucose into the blood. The result, severe hypoglycemia, especially during fasting, and the liver gets enlarged because it's just stuffed with glycogen it can't export. The bank manager can't open the vault.
Okay, I see. And another example.
McArdle disease, GSD type V.
Yeah.
Here, the problem is in muscle's glycogen phosphorylase, the enzyme that starts glycogen breakdown in muscle.
So the muscle can't access its own fuel stash.
Exactly. People with McArdle's experience significant muscle pain, cramps, and fatigue during exercise because their muscles can't tap into that quick glycogen energy source. It really highlights how vital each specific enzyme is in its specific location.
Okay, so liver glycogen gets us through maybe 8, 10, 12 hours, but fasts can last longer than that. What happens when the glycogen runs out completely? The brain still needs glucose, right? It hasn't suddenly learned to use fat.
Correct. The brain is still demanding glucose. This is when the body has to shift gears and fire up its emergency glucose factory.
Emergency glucose factory, I like that. What's the technical term?
Gluconeogenesis, G&G for short. It literally means making new glucose.
Ah, neo for new, genesis for creation. Makes sense.
Precisely. This kicks in seriously after that 8-10 hour mark, mainly in the liver, but also a bit in the kidney cortex. The body stops relying on stored glucose and starts building glucose from other non-carbohydrate molecules.
What kind of molecules? Where does it get the building blocks?
Good question. It uses things like lactate, which comes back from muscles during exercise. That's the Cori cycle. It uses glycerol, which is released when fat is broken down.
Yeah.
Pyruvate, of course. And certain amino acids, especially alanine, that come from protein breakdown, mostly muscle protein. Also things like galactose and fructose can feed in. These are the raw materials.
Okay, so it's scavenging parts from fats and proteins mainly. Now, you said glycogen synthesis and breakdown weren't simple reversals. Is gluconeogenesis just glycolysis running backwards?
Ah, another crucial point. No, it's not just glycolysis in reverse. Many of the steps are reversible and use the same enzymes, But there are 4 key steps in glycolysis that are essentially irreversible.
Okay.
Gluconeogenesis has to bypass these irreversible steps using different unique enzymes. It's like needing to find a detour around a one-way street. It takes a different route and it actually costs more energy.
What are these bypass enzymes? Any key ones to remember?
Definitely. Phosphenylpyruvate carboxykinase, usually called PPCK, is a big one. It gets around a major irreversible step early in glycolysis. Fructose 1,6-bisphosphatase bypasses another. And then, right at the end, glucose 6-phosphatase, that same enzyme we talked about in the liver, is needed to release the newly made glucose into the blood.
So that glucose 6-phosphatase is key for both glycogen takedown and gluconeogenesis in the liver.
Exactly. It's the gatekeeper for releasing glucose into circulation from the liver, regardless of whether it came from glycogen or was newly synthesized.
And how is this emergency factory regulated? Same hormones?
Very similar logic, yes.
Yeah.
Glucagon, cortisol, adrenaline, those we need energy now, signals they stimulate gluconeogenesis. They generally increase the amount or activity of key enzymes like PPCK. They turn the factory production up.
And insulin.
Insulin, signaling we have plenty of fuel, does the opposite. It decreases the levels or activity of those key enzymes, putting the brakes on gluconeogenesis. This balance is so important clinically. In type 1 diabetes, for example, there's no insulin. In type 2, there's insulin resistance. In both cases, gluconeogenesis isn't properly suppressed even when blood sugar is high. The liver just keeps churning out glucose, making hyperglycemia much worse.
Right. That makes sense. Now, let's pose another question. We're fasting long-term, glycogen's gone, gluconeogenesis is running. We have tons of energy stored as fat, right? Those massive reserves.
Huge reserves.
So why doesn't the body just take those fatty acids and directly convert them into glucose? It seems like the most obvious thing to do, doesn't it? Feed the brain directly from our biggest energy store.
It does seem logical, but it's a question worth holding on to as we talk about fats next.
Okay, let's talk fat. Long-term savings account, lipids, or specifically triacylglycerols, tags.
The ultimate energy storage.
Yeah, if glycogen is quick cash, tags are like, I don't know, high-yield bonds. You need more effort to access them, but the payout is way bigger.
That's a good way to put it. Gram for gram, fat holds more than twice the energy of carbs or protein.
And a key reason is they're stored dry, right? No water weight.
Exactly. They're hydrophobic. They repel water. So they pack together very tightly without associated water, unlike glycogen, which holds quite a bit of water. That's why they're stored in specialized cells, adipocytes, making up white adipose tissue.
And we dip into this account during prolonged exercise, stress, definitely starvation.
Pregnancy, too. Anytime energy demand is high and sustained, and the readily available carbs are running low.
So how do the fats we eat get into this storage?
It's quite a journey. In your small intestine, pancreatic lipase breaks down dietary tags into fatty acids and glycerol. These smaller pieces get absorbed by intestinal cells where they're immediately reassembled back into tags.
Huh. Why break them down just to rebuild them?
Seems odd, but it's about absorption and packaging. Once rebuilt, these tags are packaged into large particles called chylomicrons. These are too big for blood capillaries, so they enter the lymphatic system first, then eventually reach the bloodstream.
Okay, and then?
In the blood, an enzyme on the surface of capillary walls, called lipoprotein lipase, or LPL, grabs onto the chylomicrons. LPL breaks down the tags again, releasing fatty acids and glycerol. These can then enter nearby tissues like muscle for energy, or adipose tissue to be stored.
So break down, absorb, rebuild, package, transport, break down again, and then store or use. Wow.
It's efficient, if complex.
Okay, so that's storage. How do we get money out of the fat savings account? Mobilization or lipolysis?
Right, lipolysis. This happens inside the adipose cells. Stored tags are broken down by specific enzymes. The key ones are hormone-sensitive lipase, HSL, and adipose triacylglycerol lipase, ATGL.
Hormone-sensitive, so hormones control this too.
Absolutely. Glucagon and adrenaline strike again. They activate HSL and ATGL through phosphorylation, signaling release the reserves. The tags are broken down into free fatty acids and glycerol.
And what happens to those pieces?
The free fatty acids leave the fat cell, bind to albumin protein in the blood for transport, and travel to tissues like muscle, heart, liver, where they undergo beta oxidation to generate a lot of ATP.
Okay, so fatty acids for energy. What about the glycerol?
The glycerol backbone travels to the liver. And here's a key point.
Yeah.
The liver can use that glycerol. It can feed it into gluconeogenesis to make new glucose.
So part of the fat molecule can become glucose, just not the fatty acids themselves.
Precisely. And just as glucagon stimulates lipolysis, insulin inhibits it, Insulin promotes dephosphorylation, shutting down HSL and ATGL, telling the fat cell, hold on to your reserves, store more.
Okay, that covers getting energy out. What about putting energy in via synthesis,
Right.
making fat lipogenesis?
Lipogenesis. This happens mainly in the liver, but also in adipose tissue itself. It occurs when you have excess energy, particularly from carbohydrates.
So eating too much sugar or starch gets turned into fat.
Essentially, yes. The liver takes excess glucose, runs it through glycolysis to make pyruvate. Pyruvate enters the mitochondria, gets converted to acetyl-CoA. This acetyl-CoA then needs to get out of the mitochondria into the cytoplasm, where fatty acid synthesis happens.
How does it get out?
It's converted to citrate, which can cross the mitochondrial membrane. Once in the cytoplasm, citrate is cleaved back into acetyl-CoA.
Okay, so acetyl-CoA is the main building block.
It is. The first and really the key regulatory step is converting acetyl-CoA into malonyl-CoA. This is done by the enzyme acetyl-CoA carboxylase, or ACC. This step commits the carbons to fatty acid synthesis.
ACC, got it.
Then a large enzyme complex called fatty acid synthase takes over. It repeatedly adds 2 carbon units derived from malonyl-CoA onto a growing fatty acid chain. It requires energy, both ATP, and a reducing agent called NADPH, which comes partly from the pentose phosphate pathway.
So it's an energy-intensive process, too.
Very much so. To make just 1 molecule of palmitic acid, a common 16-carbon fatty acid, you need 8 acetyl-CoA units, 14 NADPH, and 7 ATP.
In regulation. Is ACC the main control point?
Yes, ACC is the star regulator. Insulin activates ACC, usually by promoting its dephosphorylation. Makes sense, insulin means high energy, so store the excess as fat. Citrate, the molecule that carries acetyl-CoA out of the mitochondria, also allosterically activates ACC. High citrate signals lots of building blocks available.
And inhibition. Glucagon and adrenaline again?
You got it. Glucagon and adrenaline lead to phosphorylation and inactivation of ACC. Low energy signals stop making fat. Also, AMP, the low energy signal we'll talk more about, and the end products, fatty acids themselves, can inhibit ACC. Feedback inhibition.
Okay. And once the liver makes these fatty acids, what happens?
They're esterified back onto a glycerol backbone to make tags. These tags are then packaged into different lipoprotein particles called VLDL, very low-density lipoprotein.
Like chylomicrons for dietary fat, VLDLs are for liver-made fat.
Exactly. VLDLs are the liver's delivery trucks, transporting newly made tags through the blood to adipose tissue for long-term storage or to muscle for use.
And this pathway is clinically relevant too, right? Obesity?
Hugely relevant. Excessive carbohydrate intake leading to high rates of lipogenesis contributes significantly to obesity, fatty liver disease, and is strongly linked to insulin resistance, type 2 diabetes, and cardiovascular disease.
Okay, so let's revisit that question. Can we make glucose directly from fatty acids? You hinted the answer was no.
That's correct. For humans, generally no. Not in a net sense. Remember, fatty acid breakdown, beta oxidation, produces acetyl-CoA, lots of it.
Right.
And gluconeogenesis needs precursors like pyruvate or oxaloacetate to make glucose. The problem is the reaction that converts pyruvate into acetyl-CoA, catalyzed by the pyruvate dehydrogenase complex, is effectively irreversible in humans.
Ah, the one-way street again.
Exactly. You can't take the 2 carbons from the acetyl-CoA and turn them back into pyruvate or oxaloacetate to make net new glucose. Those carbons from fat-derived acetyl-CoA either enter the Krebs cycle and get oxidized to CO2, or they can be used to make ketone bodies, but not glucose. The glycerol part, yes, but the fatty acids themselves, no net glucose.
That's a really fundamental point. We can burn fat for energy everywhere, except maybe the brain under normal conditions, but we can't turn it back into the glucose the brain needs.
Precisely. It highlights the brain's reliance on glucose and why maintaining blood glucose via glycogenolysis and then gluconeogenesis is so critical during fasting.
And it also highlights, again, how building and breaking down are separate paths. Fat synthesis versus fat oxidation. Completely different processes.
Totally different. Different enzymes, different locations in the cell, synthesis in cytoplasm, oxidation in mitochondria, different cofactors, NADPH used in synthesis, NADH, FADH2 produced in oxidation, and opposing regulation by hormones.
What's the advantage of keeping them so separate?
Flexibility, much tighter regulation is possible, and it avoids those energy-wasting futile cycles. Plus, it overcomes thermodynamic hurdles like that irreversible pyruvate to acetyl-CoA step. It allows the body to manage fuel flow much more effectively.
Okay, this is complex. Is there anything that helps coordinate all these different pathways? Storing glucose, releasing glucose, making glucose, burning fat, storing fat. Feels like there needs to be an overall energy sensor.
There is indeed. A really crucial one at the cellular level is AMPK.
AMPK. What's that?
Think of AMPK as the cell's internal fuel gauge, or maybe its highly sensitive accountant constantly checking the energy books.
Okay, the cell's accountant. I like that.
This stands for AMP-activated protein kinase.
Yeah.
Its main trigger is a rise in the level of AMP within the cell.
AMP, that's like ADP, but with one less phosphate, a signal of low energy.
Exactly. When ATP gets used up, you get ADP and then AMP. A high AMP to ATP ratio is a clear distress signal. Energy reserves are dangerously low. Even a small increase in AMP causes a big jump in AMPK activity.
So when the cell's running out of cash, AMPK wakes up. What does it do?
It acts like a financial advisor in tough times. Its main goal is to restore energy balance. It does this by promoting pathways that generate ATP, catabolism, and inhibiting pathways that consume ATP, anabolism.
Makes sense. Conserve resources, increase income.
Precisely. So AMPK stimulates glucose uptake into cells, ramps up glycolysis, burning glucose, and strongly promotes fatty acid oxidation, burning fat. Get energy now.
And what does it shut down?
It shuts down the big energy consumers.
Yeah.
It inhibits cholesterol synthesis. It inhibits fatty acid synthesis, remember ACC. AMPK phosphorylates and inactivates ACC. It also puts the brakes on hepatic gluconeogenesis because making new glucose is very energy expensive.
So it fine-tunes all these pathways based on the cell's immediate energy status.
Yes, it's a master regulator, integrating signals and adjusting metabolic flux accordingly. It influences glucose transporters, key glycolytic enzymes, enzymes in fat metabolism, cholesterol synthesis. It's incredibly important.
And clinically relevant.
Very much so. AMPK is a major target for the diabetes drug metformin. Understanding how AMPK works opens up potential therapeutic avenues for a whole range of metabolic disorders, from diabetes to obesity, maybe even cancer.
Wow, what a journey through the body's energy systems. It really is intricate. We've gone from the quick cache of glycogen
Stored in the liver and muscle.
to the deep savings of triglycerols in fat tissue. We looked at how they're built, how they're broken down,
Glycogenesis, glycogenolysis, lipogenesis, lipolysis.
how the body makes new glucose when needed via gluconeogenesis, and how it's all regulated by hormones like insulin and glucagon and this internal sensor, AMPK.
It's all about balance and interconnectedness. Understanding these pathways isn't just rote memorization for an exam. It's about appreciating the body's incredible adaptability, how it ensures survival, how it keeps that critical glucose flowing to the brain no matter what the energy demands are. It gives you a framework for thinking about metabolic health and disease.
Absolutely. It's fundamental. So as we wrap up this deep dive, here's something to think about. We've seen the complexity, the backup systems, the tight regulation. Given all that and how vital glucose is, what do you think the next frontier might be in managing metabolic diseases? Are we moving beyond targeting single enzymes or hormones towards maybe more integrated whole system approaches? What could that even look like? That's a big question. Definitely something to ponder.
Food for thought. Thank you so much for joining us on this deep dive into your body's fuel management system.